Students of class 11, myself Subhas Misra. Students, since it is my first video for you, so today we will first discuss about its syllabus and then we will try to complete one of the experiment. Students, your syllabus comprise 4 parts. One part is introduction, second part is core experiments, third part is experiments for spotting, and the fourth part is investigatory projects. If you see, then total number of 23 practicals are there which we have to perform throughout the year. Now the first part is introduction or introductory. This part is introductory. In this part we used to introduce you with the different equipments of lab. So this I will show you when you will come in the lab. Now its second part is the core experiment. So we are going to start from the core experiments. The first experiments which today I am going to conduct, that is related with osmosis. So students, first of all, do you know what is osmosis? Of course, osmosis is the process in which movement of solvent particles takes place from higher concentration to lower concentration. It is of two types, X osmosis and end osmosis. End osmosis, in which the solute particles, they move from outside to inside the cell. And X osmosis, where the movement of solvent particles takes place from cell to outside the body. Students, why this transfer of solvent particles takes place? This is due to the difference in the concentration of the solvent particles. So you can understand it very well by means of an example. You have already seen that when you put reagents inside the water, the reagents get swell up after some time. Why do they get swell up? Because they have absorbed water from outside. That is, the concentration of water inside the reagent was less as compared to the outside. That is why water molecules move from higher concentration to lower concentration. That is, from that place where their quantity is more as compared to the place where its quantity is less. So they move from outside to inside the reagents. This is end osmosis. Now X osmosis is if you take out the reagents, put it on the table and leave it for few hours. You will see that reagents get shrink. Why it happens? Because the solvent particles, they move from reagents to outside the surrounding. Now why it happens? This is also due to the difference in the concentration, or you can say the concentration of water molecules was more inside the reagents as compared to outside the surrounding. That's why it moves from reagents to outside. Here the skin of reagents, it acts as semi-permeable membrane. So in this way, osmosis is the process where transfer of solvent particles takes place from higher concentration to lower concentration through a semi-permeable membrane. This is osmosis. Now osmosis in the lab, we are going to find out this activity in the lab by means of osmometer. What is the benefit of osmosis or its function? Turgidity of plants. It is due to osmosis process. Opening and closing of stomata. This is due to osmosis. So likewise, it has lot of functions. Now, factors on which it depends. Osmosis depend on temperature and the particles, number of particles or concentration of particles in the solution. So these are the things on which osmosis depend. In the next video, I will give you the whole procedure how we are going to perform the experiment.